So the next experiment to find out that the DNA is a genetic material is the Hershey and Chase's experiment. These were the two scientists who did their experiment to find out, to give the idea that DNA is the genetic material. It is also called bacteriophage multiplication or transduction. Why it is called bacteriophage multiplication? Actually bacteriophage, phage word is generally used for virus. Bacteriophage is a bacterial virus which infects bacteria. जैसे वायरस हमें इन्फेक्ट करते हैं, हमें इन्फेक्ट करते हैं, उसी तरह से दे कैन ऑल्सो एंटर इनटू बैक्टीरियाज. ठीक है. एग्जाम्पल इज ही डिड हिज एक्सपेरिमेंट ऑन T2 बैक्टीरियोफेज, जो कि एक वायरस है, बैक्टीरियोफेज वायरस, जो कि किसको इन्फेक्ट करता है? ई कोलाई बैक्टीरिया, एस्टीरिचिया कोलाई बैक्टीरिया को इन्फेक्ट करता है. और इस प्रोसेस को हम क्या बोलते हैं? ट्रांसडक्शन. जब भी एक वायरस बैक्टीरिया को इन्फेक्ट करता है, तो अगर हम वायरस का स्ट्रक्चर देखें तो इसके अंदर क्या होता है? इस तरह से प्रोटीन कोट होता है, बाहर प्रोटीन की लेयर होती है और अंदर इसका जेनेटिक मटेरियल होता है. डीएनए भी हो सकता है या आरएनए भी हो सकता है. ठीक है. तो जब भी ये वायरस जो होता है किसी भी बैक्टीरिया को इन्फेक्ट करता है, तो ये अपने सिर्फ जेनेटिक मटेरियल को ट्रांसफ़र करता है, प्रोटीन को बाहर छोड़ देता है. ठीक है. इस प्रोसेस को हम ट्रांसडक्शन बोलते हैं, इसीलिए यहाँ पे हमने इसको ट्रांसडक्शन भी बोला है. तो ही डिड हिज एक्सपेरिमेंट बाय यूजिंग द T2 बैक्टीरियोफेज, दैट इज़ अ वायरस हैविंग डीएनए एज जेनेटिक मटेरियल. क्योंकि वायरस भी दो टाइप के होते हैं, एक डीऑक्सीराइबोवायरस एंड राइबोवायरस. डीऑक्सीराइबोवायरस जिनमें डीएनए जेनेटिक मटेरियल होता है और राइबोवायरस जिनमें आरएनए जेनेटिक मटेरियल होता है. तो इट इज़ अ डीऑक्सीराइबोवायरस. तो आपको पता है कि जो डीएनए होता है, जब हम इसका डिटेल स्ट्रक्चर स्टडी करेंगे, इसके अंदर नाइट्रोजनस बेस, फॉस्फोरस, शुगर, ये सब प्रेजेंट होता है. ठीक है. तो उसमें से इसने क्या किया? दो रेडियो एक्टिव आइसोटॉप ले लिए. रेडियो एक्टिव आइसोटॉप लेने का पर्पज़ ये होता है कि जब हम स्टडी करते हैं तो हमें वो बैक्टीरिया जिसके अंदर रेडियो एक्टिव आइसोटॉप्स होते हैं, रेडियोलॉजी के थ्रू इजीली एक्सरे के अंदर बहुत क्लियरली दिखाई दे जाते हैं. अदरवाइज़ हमें पता ही नहीं चलेगा कि वायरस अंदर गया है या नहीं गया है. ठीक है. तो इसने क्या किया? दो रेडियो एक्टिव आइसोटॉप लिए, फॉस्फोरस का और सल्फर का. फॉस्फोरस वॉज P32 एंड सल्फर वॉज P35. ओके. And then he did his experiment. Let's see the experiment. How did he do his experiment? So if you will see the Hershey and Chase experiment, what they did, I have told you, they have taken two radioactive isotopes. One was the sulfur, that was the component of protein coat, because मैंने आपको बताया था बैक्टीरियोफेज जो है उसके बाहर एक प्रोटीन कोट होती है, अंदर डीएनए होता है. तो सल्फर वॉज प्रेजेंट इन द प्रोटीन कोट एंड डीएनए के अंदर जो रेडियो एक्टिव आइसोटॉप था, दैट वाज फॉस्फोरस 32. ठीक है. क्लियर है. उसने दो केसेस लिए. इन फर्स्ट केस ही टुक प्रोटीन कोट एज रेडियो एक्टिव, इन द सेकेंड केस ही टुक डीएनए एज रेडियो एक्टिव मटीरियल. और जो मैंने आपको बताया था कि वेन एवर दिस बैक्टीरियोफेज, कोई भी बैक्टीरियोफेज जब बैक्टीरिया को इन्फेक्ट करता है तो ये अपने डीएनए को अंदर रिलीज़ कर देता है, प्रोटीन बाहर रह जाता है. लेकिन उस टाइम पे साइंटिस्ट को ये बात नहीं पता थी. That is why he took two different radioactive isotopes, one in the protein coat and one in the DNA. उसके बाद इसने क्या किया? दोनों एक्सपेरिमेंट साथ साथ किए और मिक्स कर दिया, शेक किया. एंड देन उसके बाद क्या होता है? जो बैक्टीरियाज होते हैं वो अपने होस्ट सेल की मशीनरी को यूज़ करके अपना पूरा स्ट्रक्चर बना लेते हैं. प्रोटीन बना लेंगे और साथ में अपना जेनेटिक मटेरियल बना लेंगे. तो दोनों केसेस में यहाँ पे प्रोटीन इसने बना लिया, अपना प्रोटीन कोट बाहर से जो मैंने आपको हेक्सागोनल शेप दिखाया है, एंड जेनेटिक मटेरियल, दोनों चीज़ें इन्होंने प्रिपेयर कर ली. आफ्टर दैट, आफ्टर फॉर्मिंग इट्स स्ट्रक्चर, द बैक्टीरियोफेज ब्रेक ऑफ द बैक्टीरिया को ब्रेक करके बाहर निकल जाते हैं. ठीक है. उसके बाद फिर इन्होंने क्या किया? इसको आइसोलेट किया और स्टडी किया एक्सरे के अंडर. देन दे फाउंड दैट द बैक्टीरियाज वर कम्प्लीटली द वायरसेस वर कम्प्लीटली नॉन रेडियो एक्टिव. उन्होंने देखा जो बैक्टीरियोफेज ई कोलाई से रेप्लिकेट होकर निकल के आए, रिप्रोड्यूस होकर निकल के आए, वो कम्प्लीटली नॉन रेडियो एक्टिव थे. और इस केस में जब उन्होंने देखा बैक्टीरियाज को एक्सरे के अंडर, तो देखा वायरसेस को, तो दे वर कम्प्लीटली रेडियो एक्टिव. सो दे केम टू नो दैट देयर इज समथिंग इन द प्रोटीन कोट. When the protein coat was radioactive, the viruses which were coming from the E. coli were non-radioactive. But when the DNA was radioactive and the viruses which were coming out from the E. coli was radioactive, it means that they came to know protein coat does not have any role to transfer the information into next generation. DNA is the only material which will transfer the information to next generation. क्योंकि इससे उनको क्लियर हुआ प्रोटीन कोड तो बाहर ही रह गया था, सिर्फ डीएनए अंदर गया था, और यहाँ पे डीएनए के अंदर कोई रेडियो एक्टिविटी थी नहीं, यहाँ पे डीएनए के अंदर रेडियो एक्टिविटी थी. तो जब ये वाला रेडियो एक्टिव डीएनए अंदर गया और यहाँ से रेप्लिकेट होके निकला, तो नेक्स्ट जनरेशन में जो सारे वायरस बने वो सभी रेडियो एक्टिव थे. इट मीन्स दैट डीएनए ही ऐसा मटेरियल है सेल के अंदर जिसके अंदर जो भी फीचर होंगे वो नेक्स्ट जनरेशन में ट्रांसफ़र हो जाएंगे. अब वो फीचर्स कभी तो बिल्कुल एग्जैक्टली सेम ट्रांसफ़र हो जाते हैं और कभी उनके अंदर थोड़ा चेंज हो जाता है डिपेंडिंग अपॉन द वेरिएशन, जो हमने म्यूटेशन और वेरिएशन को स्टडी किया था. From this, he concluded that DNA is the genetic material, the molecular or the chemical basis of inheritance. So with this, the DNA, the genetic material, has been completed. Now we will see the structure in detail. Then I will show you how the other characteristics matches with the DNA structure.
And the Chase experiment was one of the most remarkable works in the field of genetics. What was the experiment about? And what does it help us infer? Let's find that out. In the timeline of the history of genetics that we looked at, we have reached this point. So let's step ahead and jump to the year 1952. Two American geneticists, Alfred Hershey and Martha Chase, carried out a very simple experiment that helped figure out whether DNA was really the transforming principle or not. The experiment was quite simple. It just required a culture medium, two different radioactive isotopes, a mixer blender, and a centrifuge tube. A culture medium is a medium used to grow different kinds of microorganisms. With this equipment, they carried out an experiment using a bacteriophage and a bacterial strain. Let's find out how the experiment was framed. Before that, can you tell me what bacteriophage is? The term phage means virus, so the name bacteriophage stands for virus that infects bacteria. So bacteriophages usually infect the host bacterium by inserting their genetic material into them. And they make copies of themselves using the replication machinery of the host bacterium. The interesting thing here is that the body of any bacteriophage is simply made up of two components, the outer protein coat and the inner genetic material. Now, let's get back to the Hershey and Chase experiment. The main aim of their experiment was to find out what exactly is the genetic material. Is it proteins or DNA? Since the phage contains only proteins and DNA, it's easier to find out which component gets into the host bacterium. They simply used radio-labeled markers to identify the different strains. So if the bacterial cells have the radioactively marked proteins in the end, then it's the proteins which are the genetic material. On the other hand, if the radio-labeled DNA makes its way into the bacterial cell, then probably the DNA is the genetic material. So the experiment was quite simple. They first obtained radio-labeled bacteriophages. One strain had the radio-labeled proteins and the other had radio-labeled DNA. Then they allowed these different bacteriophages to separately infect the bacteria. And at the end, they analyzed the bacteria to find out which radio-labeled component had made its way into it. But how did they obtain the radio-labeled proteins and DNA? What was the process they used? And how were the results analyzed at the end? Let's find that out in the next part. We know that their bodies are made up of only proteins and DNA. That makes it easier to figure out what gets into the host bacterial cells. The experiment was quite simple. They first obtained radio-labeled bacteriophages. There were two different types, ones with radio-labeled proteins and the others with radio-labeled DNA. Then they allowed these different bacteriophages to separately infect the bacteria. And at the end, they analyzed the bacteria as to which radio-labeled component had made its way into it. This is how the simple experiment helped answer a few important queries. Now let's get into the details of this experiment and understand it step by step. The first thing Hershey and Chase did was that they obtained the radio-labeled phage strains. In one nutrient medium, they added the radioisotope sulfur-35, which is S35. In the other, they added phosphorus-32, which is P32. But why only sulfur and phosphorus? And that too in separate mediums? Well, sulfur is an integral part of proteins, but it's not present in the DNA. On the other hand, phosphorus is present in the DNA, but not in the proteins. Thus, for easy analysis, they chose the radioisotopes of these two elements. So one nutrient medium was added with the sulfur-35 radioisotope. The bacteria grown in this medium does had S35 incorporated into their bodies. Now, what will happen if the bacteriophages are allowed to infect these bacteria? It's obvious that S35 will get incorporated in the protein coats of these bacteriophages as well. That is because these are derived from the body of these host bacteria. So their first batch of S35 labeled bacteriophages was ready. Similarly, in this culture medium, they added P32 and allowed the growth of bacterial colonies. As a result, all the bacteriophages obtained post-infection had radio-labeled DNA. So the second batch of bacteriophages with radio-labeled DNA was also ready with them. Now, all they had to do is allow these radioactively marked bacteriophages to infect new bacterial cells. These host cells will be the ones harboring the new phage generations, which will help analyze what has passed down the line. So they infected the bacterial cells with the two batches separately. After several hours of infection, the cultures were agitated in a mixer blender. Why is this step necessary? It is needed to make sure that post-infection, the empty phage coats get separated from the bacterial cells. The agitation helps release the empty phage coats attached to the bacterial cells. Now, in the next step, they centrifuged the culture at an extremely high speed. Centrifugation process works on the principle of gravitational force. So when the sample is centrifuged at a very high speed, the heavier, denser particles settle at the bottom, while the lighter particles remain on top. What is settled at the bottom is usually called pellet, while the one that floats on top is called supernatant. So the centrifugation step helped Hershey and Chase to obtain two layers. The one on top was supernatant, and the one at the bottom was pellet. They checked the radioactivity in both the batches and concluded their analysis. Before we reach the conclusions, can you help Nora guess the interpretation? What will be the interpretation if the radioactivity is found in the supernatant? And what will be the inference if it's found in the pellet? Think about it. Let me help you with this. The supernatant contains phage hosts, which are the empty shells or coats of phage. It does not have the replicated next generations of the bacteriophages. Similarly, the pellet will contain the bacterial cells and the newer phage particles. So now, in case 1, if the radioactivity is observed in the supernatant, then it means that the marker is only present in the protein coat of the phage and did not get into the bacterial cells. This can help us conclude that the proteins are not the genetic material, as the newly formed phage particles did not receive S35. On the other hand, if the radioactivity is seen in the pellet, then it would mean that the newer phage cells have all got the radioactive P32 in them. Thus, the DNA in the form of genetic material has successfully got transformed into the new phage particles. This is what the analysis of both the cases can be. Now let's look at the results Hershey and Chase obtained. On analyzing the first sample, They found that the radioactivity was present in the supernatant. And in the second batch, the radioactivity was found in the pellet. What did this mean? That's right. With this, the two concluded that it's the DNA which is the genetic material and not the proteins. If proteins would have been the genetic material, then the pellet in the first batch would have shown the presence of radioactive S35. Instead, the pellet in the second batch showed the presence of P32. Thus, it was confirmed that it's the DNA which is the genetic material. With this, Avery, MacLeod, and McCarty's analysis that Griffith's transforming principle was DNA was also proved. Thus, the long-ignored DNA molecule now rose to fame, and scientists all over the globe started working on finding its structure. But how was the structure discovered? And what does the actual structure of a DNA molecule look like?